The SHIP study before, and I'm very sure we're listening to some more SHIP today. And Johannes Hertel started out at Kiel, just to make a connection here. Kiel and Lübeck are not that far from each other. And he also spent a year in Luxembourg on his career and Ireland. And since 2014, he is working at the Universitätsmedizin of Greifswald in the institution of the Department of Psychiatry. So, very welcome. And your talk is on epidemiological approach to clinical NMR applications.
Yeah, thank you very much for the possibility to talk here. And yeah, I'm the last person in a long day. And now it comes to statistics because I'm a statistician. So, but I hope I will keep it on a high level so you can follow what is done here. And for me, like we are living really in exciting times because we are developing so many measurement technologies and we can quantify the human body in a detail which is really unprecedented. But then the question is, yeah, what do we do with all the data? And somehow we need to transfer all this data into mathematical representations where the dependency terms, like stochastic dependency or so, represent like the causality in the biological systems. And then we need to translate once again the mathematical dependency terms and like clinical decision rules. So for me as a mathematician, as a statistician, actually this is a mathematical problem, yeah? So how do we find mathematical structures which represent the biology in a way that allows us to make this kind of translation for individualized medicine? And I want to talk about Nmr Metabolomics today because, yeah, from all the Omics data actually I have seen in my life, Nmr Metabolomics is still one of my favorites. And for some really good reasons. And I want to showcase two examples of my work with NMR, urine metabolomics in this case. One is measuring biological age in the SHIP study, and the second one is describing microbiome-host interactions. Yeah, and as already said, all these results come from a SHIP study, which we already learned about a little bit. So a beautiful longitudinal cohort study with a lot of metadata and omics data attached. All right, so what we really need to achieve is somehow to get from all the omics data to individualized phenotypes. And the problem here is somewhat that our clinical phenotypes, they have grown historically over the last 150 years in the ICD-10, and they are not designed to reflect individual biology. So we have a problem that our outcomes, our clinical outcomes like age, chronological age, or BMI, or diabetes, they are rather non-individualized. So the question is, can we use omics data to achieve an individualization of those non-individualized clinical phenotypes? And yeah, in my PhD work, actually, I came up somewhat with a data-driven procedure. However, was also a little bit negative because to make this work, one really need to understand the source of stochastic dependencies in omics data. And when you think about why do two metabolites correlate in the urine, there are a lot of sources of this correlation. And that makes this kind of step difficult. Still, I think we can achieve something, and I want to highlight this with the example of biological age. And actually, biological age is exactly a problem of individualization because two persons of the same chronological age, like 70, they may have different biological ages due to genetics, due to environmental impacts, due to lifestyle. So what we do is, like in the data set we're working with, we assign the same phenotype, the same chronological ages, but actually there's a different underlying biology. And what we would like to have is like a true measurement of biological age that reflects different health risks accordingly. So how can we conceptualize this mathematically? Actually, this is something which is probably not too far away from analytical chemistry thinking because it's a measurement error problem. Yeah, the chronological age is somewhat a measurement of biological age, but it has a measurement error. So what we can do is like apply these kind of standard measurement error models to get a mathematical grip on the problem. for example, we could say biological age is chronological age plus some error. Unrelated chronological age, which would be the Bergson error measurement error model, or the classical measurement error would be chronological age is biological age plus some error unrelated to biological age. And in this case, I feel somehow that the Bergson measurement error model is more intuitive and more related. So what we are interested actually is the difference between biological age and chronological age. This is the one thing we want to find out. Yeah? If we would have this different, so the Bergson error, then we would have a perfect quantification of biological age. Now we need some kind of tool how to estimate it. And yeah, I know there are a lot of papers out there measuring biological age using epigenetics or any kind of omics data, basically, and we can use the urine metabolome as well. And what all these papers do, and what also I did, was like we predict via some kind of machine learning the chronological age, in this case via urine metabolome data, targeted urine metabolomics in the SHIP study, where we had like 60 measurements, 60 different metabolites, and then claim that the prediction score is somewhat an estimate of biological age. And that's somewhat weird, because we predict chronological age and then claim it's a measurement of biological age, yeah? So this sounds non-plausible, but all their epigenetic clocks and so on, they make exactly the same step. They predict chronological age, and then they claim, oh, our score has some kind of prediction error, and this prediction error is the Bergson error, and a person who has a higher metabolic age score, or higher epigenetic clock, has a higher biological age. And interestingly, it always works somehow, and it works here as well. So the people who have a higher biological metabolic age score than their chronological age, they have lesser survival time. So in the SHIP study, we have survival data, so we follow the people over time. They have a higher burden of comorbidities, age-related. So it works pretty well. Perhaps one point regarding this estimation procedure to predict chronological age, it's really, really important that this is sex-specific and nonlinear, because the age trajectory of urine metabolites, when if you have a sample like SHIP, where you have 20 to 90 or 20 to 80 years, it's not the same for women and men, because men, for example, don't have a menopause. So all the trajectories over age look really completely different, and you have certain metabolites where men have no trajectory at all, and women have like this. So it's really important, if you do machine learning on such data sets, and if you are interested in those kind of phenotypes like metabolic age or biological age, you need really to think about sex specificity and nonlinearities, because otherwise you will produce biased results. Now let's go a little bit deeper in the results. So because SHIP is such a beautiful cohort, we can not only look at survival, but we can also look at various other markers of metabolic health. And basically, people who are physically active are younger in the metabolic age score. People who have diabetes are older. People who have like liver, kidney dysfunctions like albuminuria are older, and so on. So a lot of age-related phenotypes are actually associated with a higher biological age on this metabolic age score. So it works pretty well. And interestingly, and that's something where I feel that the benefit of NMR really, really comes in. I utilized the same prediction score, really the same, numerical, the same prediction score on 3 different cohorts, SHIP trend 0, SHIP 0, these are 2 different cohorts of the SHIP cohort, and then a small sample with bariatric surgery. And in all these 3 samples, this prediction score replicated to a certain extent. And this is something you would not get with mass spec data, because in mass spec data you have always the problem of batch effects and so on. So, but like with MNR, you get very robust results. And for clinical applications, this is really, really a benefit, because if you have like complex machine learning and then something small is off in the covariance matrix, your results are off. And with NMR, you have the stability to actually make this work. Now, this actually is an example of a generalized framework for individualization. So you could use the same procedure not only for biological age, but let's say for healthy obesity. So you use measurement of obesity, like waist circumference. You predict the waist circumference with some kind of machine learning model, and then say those people with a higher metabolic waist circumference than the real waist circumference, those are the unhealthy obese. And then there are also healthy obese. Or you predict diabetes via machine learning, and then you have misclassification, and you say, okay, those misclassifications are actually informative, because those who are misclassified in the prediction algorithm probably are more healthy than those who are correctly classified, for example. So what you do is you predict the phenotype, then you regress technically the prediction score on the phenotype, deriving the residual variable, the error term, and then you demonstrate that this error term has some informational content, like we did here in the question of biological age. Now, the question is, why does this work? Because it sounds a little bit like voodoo, but no, it's actually mathematics, and one can prove that under certain stochastic constraints, this kind of procedure will always work. However, those kind of constraints you have to put in are really, really strong, so it's also a little bit of luck that this works so well. Okay, and then one thing I want also to mention is this is the best H-score, like looks very convincing, replicated on 3 samples, predicting all kinds of comorbidities. Is this the best H-score we can get given the urine metabolome? I'm pretty sure it's not. And I'm pretty sure that even on the SHIP study, one could come up with a better score. So I have the feeling that when it comes to quantifying biological age via NMR metabolomics, there's still a lot to discover. All right, so this was the first example. Now to something completely else. We all heard about the gut microbiome. It's very en vogue these times. Because now we have ways to measure the composition of the gut microbiome by sequencing of fecal samples. And the gut microbiome is obviously a very essential part of our bodies because we have evolved in the world of microbes, and there are a ton of studies showing that the microbiome shapes the immune system, contributes to metabolic health, contributes to neurodevelopment, the gut-brain axis. So the gut microbiome is really important for human health and disease. And what people are doing is, they use sequencing data to quantify the relative abundances of taxonomic units like strains, species, or genera in the fecal samples. And then they have like the composition of the microbiome for each person, and then they compare how those compositions are structured in healthy and diseased people. But the problem with this approach is that, microbes, those are different strains and different species, but they share a lot of their metabolic capabilities. So you can have two totally different microbiomes, which are functionally really equivalent. So just looking at abundances and just looking at composition is just the first step. It's not the full story. So the question is, how can we analyze whether those differences and abundances are actually translating into differences in metabolic functions? And there NMR metabolomics comes in once again, because in the urine NMR metabolome there are many, many interesting metabolites, partly currently not even covered by the standard mass spec platforms. So for example, methylamine, dimethylamine, trimethylamine, methanol, format, indoxysulfate is measured by some of the mass spec platforms, but for example, formic acid, which is a standard fermentation product of bacteria, I have not seen it in any mass spec platform. So there are a lot of metabolites produced by the gut microbiome, which can be well measured by NMR and currently even exclusively in this kind of high throughput way. So now what we want to do is asking whether gut microbiome species have a association pattern in the NMR urine metabolome, and can we find correlates of gut microbiome diversity in the NMR metabolome? Because one factor which has always be on the top list of gut microbiome research is saying, if you have a diverse community in the gut microbiome, a lot of different species, that's generally beneficial for health. the question is, yeah, why is that actually the case? Why should that be the case? And one point could be that this kind of diverse communities translate in different metabolic functions. So what we tried to do here is in the SHIP trend cohort, where we have around 950 people with 16S sequencing data of fecal samples, and Nmr Metabolomics, try to find association patterns between the gut microbiome composition and the NMR metabolome. And yeah, we had a very, very clear signature with hippurate. Hippurate is a glycine conjugation of benzoate, and benzoate is a microbiome metabolite or a diet-derived metabolite, and that was like the strongest signature, which also was replicating earlier results. Then we found association with succinate. Succinate is also a main fermentation product of many bacteria. It's also like main human metabolite, so the amount of variance explained is by bacteria is low there. And we had indoxyl sulfate, which is a downstream product of indole, an end product of microbial tryptophan metabolism. And indoxyl sulfate is also interesting because it's thought it's an indicator of chronic inflammation and also an indicator of it's a uremic toxin. And we found some associations with formate, which is also a main fermentation product of many bacteria. Now I said that hippurate was the strongest signature. It also was by far the strongest correlate of microbiome diversity. So people with high diversity in the microbiome had higher urinary hippurate levels. And actually, if we ask ourselves, okay, now we do machine learning and use all the fecal data we have and try to predict the urinary metabolite concentration, that works generally not so well because, okay, human metabolism is too dominant, but for hippurate, really a substantial part of the variance is explained by the gut microbiome. So now we can ask ourselves, if higher microbiome diversity is generally a correlate of better general health, is the same true for hippurate? Yeah? Because if it's readout of gut microbiome diversity, that's a plausible question. Now, if you look, now we tested a range of standard markers like liver damage, like inflammation, and metabolic health. If you look at urinary hippurate, well, not so great, basically. If I would correct for multiple testing here, nothing would survive. If I look at the Shannon entropy, which is a measure of microbiome diversity, you actually see that there are some solid associations with markers of metabolic health and liver damage. And then if you look at the microbiome-based hippurate prediction score, you see a very strong signature. And that's interesting because like this, we used the urine metabolome to identify a component of the gut microbiome, which is apparently very predictive for certain clinical markers. So in this case, it's not the NMR data actually is not itself so predictive for health, but we used the NMR data to inform where to look in the gut microbiome. Because this kind of prediction score can be interpreted as the features in the gut microbiome which correlate most with the hippurate concentration. And actually, this prediction score completely mediates all effects of metabolic health of the Shannon entropy, so of gut microbiome diversity, which is an indicator that benzoate metabolism and benzoate microbiome co-metabolism is actually one of the factors mediating the effects of gut microbiome diversity. Maybe a short look on the pathway. What we're talking here is, yeah, we have like the human part which takes benzoate and produces hippurate, and hippurate is then secreted in the urine. And the gut microbiome either degrades benzoate or it can also produce benzoate. And we looked also up in other researches and did some functional annotations using genome scale reconstructions. And we found basically that the lower the benzoate degradation potential, the higher the diversity in microbiome communities. So this means that highly diverse communities have lower benzoate degradation potential, which means that more benzoate arrives in the human, and then you have more hipporat in the urine. So if you would ask for a potential target on the microbiome, which is always like a tricky thing, because it's an ecosystem which is hardly to predict how it reacts to intervention, then one would say, okay, what we need to do is we need to increase the microbial benzoate secretion, or we need to decrease the microbial benzoate consumption if this pathway is causal, which is of course a big assumption. So right now it's just an association. Okay, so coming to the conclusion, so NMR metabolomics can be employed in large-scale epidemiological research in a versatile way, and I really had fun, a lot of fun with NMR data, and I hope to have a lot of fun in the future. NMR metabolomics can be utilized to get insight on biological age of a person, and I think a lot of other traits as well. And prediction score from NMR metabolomics have a striking stability, which is, I think, a very important point to remember. And also, I like the way how we can use NMR metabolomics to describe host microbiome interactions. And we saw now that benzoate co-metabolism is a strong candidate for being a mediator factor between health and microbiome diversity. So yeah, a lot of people are involved in this kind of work, obviously. So yeah, and thank you for your attention.